Going. There we go. Okay. So we are continuing our way through membrane receptors and signal transduction. And when we talked at the end of last week, this is where we left off. We were talking about the adenyl cyclic AMP pathway. Just to refresh you on how this works, you start off with a ligand up here, and it's going to bind to a G protein coupled receptor that's on the cell membrane surface. That is then going to interact with this G protein, at which point this alpha subunit that has a GDP attached to it is going to unbind, and it's also going to swap its GDP for a GTP. So now it's a GTP associated alpha subunit, and it goes and it is going to activate adenyl cyclase. Adenyl cyclase then comes over here to the ATP and converts ATP to cyclic AMP. Cyclic AMP then attaches to a phosphokinase A, which activates the phosphokinase A, which then, not phosphokinase, protein kinase A. It's going to then activate this protein kinase A, which is going to phosphorylate some sort of enzyme. I'm going to talk more about what that could be later. And is going to then convert that into a phosphorylated enzyme, which activates it. Now, this isn't always the case. This could also, in some cases, deactivate it. It could turn it on. It could turn it, or turn it up. It could turn it down. So it could not totally turn on or not totally turn off. So there's a lot of things that this can do. But essentially, just remember that this is going to modulate this enzyme's activity. It's going to change it somewhat. Then this enzyme is going to either turn substrate into product, or it's going to stop turning substrate into product, depending if we have turned it on or turned it off. Are there any questions up to that point? Just kind of a quick refresher. Yeah, Alex.
Is there a functional reason why there's so many steps? Why we don't just take from the ligand to the receptor to phosphorylating something?
Yes. So this is a good one. I talked about at the end of class. Does anyone remember? Amplification. So that's a really big reason, is amplification. So actually, through these steps, we actually get about billion-fold amplification for every ligand that we get, for every ligand that we bind to the receptor up here. On average, now this is like within a few orders of magnitude, we can get about a billion product made. And this is really, really nice, because then we don't have to use much of the signal molecule to actually get tons of our product that we want to actually get out. The other thing is, each step along here, you actually get opportunities for control and kind of nice fine-tuned control if you have more steps. So there's more places that different processes can intervene and kind of modulate the effect here. So that's another kind of advantage of doing that. Does that answer your question? Awesome. Okay, so let's talk about some more things starting off with this. First off, there's a really interesting other protein that is generally attached to protein kinase A, called AKAP, or just stands for a kinase anchoring protein. Does anyone wonder what it does? It anchors a kinase. And there's a couple things. So this is nice. It holds pKa in Monday. It holds it in Monday. It holds it in specific locations in the cell. Now, if you remember back when we were talking about how we can regulate enzymes, one of the major ways that we can regulate how an enzyme functions is holding it in some specific location in a cell where it's going to be close to things that it needs to react, close to other enzymes in some sort of chain, something like this. And this allows us to have an efficient use of going from product of one enzyme to become the substrate of the next enzyme, and on so down the line. And this can also hold other enzymes close to phosphokinase or protein kinase A. I'm going to keep trying to say this wrong, so forgive me. It can also hold other enzymes. One big one that it often holds is a thing called a phosphodiesterase. A phosphodiesterase will break down cyclic AMP. Can anyone think of why you would want something next to AKAP or next to pKa that's breaking down cyclic AMP? Okay, it will get the phosphate group off, but in a bigger, but kind of more overarching, why would, okay, so yes, you can do that. Why would you want to do that?
Your phosphorylating enzymes.
Yes. That's actually generally, the phosphate doesn't come from cyclic AMP. But that's a good guess. That's a very good guess. Any other ideas? Exactly. Yeah. So you don't want to have the signal turned on forever, and so you need to turn it off. And sometimes you want these things to turn off quickly. And so having a phosphodiesterase right next to pKa can allow this to be turned off. Like you give it a cyclic AMP, and it'll turn on, but just for a moment, which means that you want to keep having this event happen. You need to keep giving it cyclic AMP to keep it on. And as soon as you stop giving it, it turns off very quickly, which could be nice. You want to be able to have fine control in time as well, so you don't want to have things like, okay, I've stopped the gas. Like imagine if you're driving a car around, and like you let off the gas, but it takes like 30 seconds for actually like the gas to lower. You don't want that to happen. Like you want the gas to drop off as soon as you let off the pedal, or else crashes, bad things. Anyhow, so yeah, this is essentially shutting it off as soon as you stop giving the signal. Now that being said, there's something like more than 50 known AKAPs out there. There's a lot of different ones. They occur in different cells. Actually, they don't just hold protein kinase A. Yeah.
So is one AKAP holding just like one kinase or multiple at the same time?
Depends on the AKAP. So some do only hold one, and some hold both. I think, if I remember right, I think the more common end is to hold multiple, but I can't say completely for sure. I'm kind of making a guess there from what I vaguely remember. But there's a lot of different ones. They occur in different cells. Actually, not all. They don't all just hold protein kinase A. There's other protein kinases that they hold. Any questions on that? Yeah, Christian.
So they hold phosphodiesterase near the pKa?
Yes.
So would it be the same AKAP that's holding the pKa that's holding?
Correct.
Okay.
Yes.
Cool.
Good questions. Any other questions?
If it's going to only hold just one PK, isn't it going to sacrifice amplification a little bit? Like, you're deactivating the cyclic AMP right away, where it would normally create 100 enzymes, but now it's only making.
Well, but the PKA, it depends on how long it goes. But, and I think that 100 enzymes is still with the phosphodiesterase there. That's in exchange for, yeah. I think generally they have a phosphodiesterase and they're still getting that much because most enzymes work really fast. So even if you shut them off very quickly, they've done well. Yeah. Good questions. Anything else? So let's take a look. What are some of the things that will happen? This system actually causes to happen. The effects of the cyclic AMP PKA enzyme complex. So a couple things that things will affect. These will enhance or increase lipolysis in adipocytes. You'll increase glycogenolysis. This is the breakdown of glycogen. So you're taking glycogen, which is a energy storage molecule, turn it back into glucose to use it for energy. You decrease glycogenesis, which makes sense. If you're breaking down glycogen, you don't want to be making glycogen again. You want to get glucose out. So you're decreasing the making of glycogen and increasing the breaking down of glycogen. And they also increase in skeletal muscle glycolysis. So this is the pathway we're going to talk about in just a moment here. I'm sure you probably talked about this in general biology, which we take glucose and get some energy out of it and actually get going, pass on the products to citric acid cycle and electron transport chain eventually. Now, in cardiac muscles, we're sequestering calcium in the sarcoplasmic reticulum. That's a long story about what's going on there. But and then in the hepatocytes, so this is in liver cells, we do some similar things to what we do up here in skeletal muscle. We're increasing glycogenolysis and decreasing the glycogenesis. And we're decreasing glycolysis. And we're increasing this thing called gluconeogenesis. This is making glucose from non-carbohydrate molecules. And then principal cells, these are actually down in, actually they're in quite a few things, but we'll come back to that. You're increasing aquaphor 2 expression, and then smooth muscle, you get relaxation in smooth muscle. Especially smooth muscle that is surrounding certain blood vessels. So I want to just back up here for a second. Just taking this all kind of together, what do you think this does? If we're increasing in skeletal muscle glycogenolysis, decreasing glycogenesis, and then increasing glycolysis, what kinds of things is the skeletal muscle getting ready for? Yes, hard work. Your skeletal muscles are essentially getting ready for some sort of work, or responding to work. Because we are breaking down our energy storage to get energy out of them. We're stopping storing energy because we want to use it, not store it. And we're increasing the production of energy from that glycogen. Okay, so if that's the case, why here in the hepatocytes do you think we're decreasing glycolysis?
But that's not muscle.
Yeah.
So it wouldn't need to work as hard. Well, you wouldn't want your organs working overtime and your muscles working overtime at the same time.
Exactly. So what's happening in the liver actually is it's mobilizing glucose, and instead of using it itself, it doesn't want to use it. It's shipping it out to the blood and getting it to your muscles. It's acting as a reservoir of glycogen so it can get it out, or reservoir of glycogen so it can turn into glucose and get it out to your muscles. So it doesn't really want to be using it because it's not like your liver flopping around is going to help you run away from a bear. That sounds weird. Or whatever livers do. I mean, that's not what livers don't generally flop on their own. Don't learn anything else. Learning that livers don't flop on their own, they're not muscles. Anyways, but the point is that we don't want the liver using a ton of energy because we don't need it using energy right now. But I want to point out again something that I kind of pointed out last week is that, again, here is one system that has completely different effects in two different areas of the body. Is in one area we're going to crank up glycogen or glycolysis, and another place we're going to crank down glycolysis, and we're using this exact same pathway. We're using this exact same thing right here. It's just these ends are different right here. And so I just want to point out that one more time, is that one nice thing about these signal cascades is also that you can get these different effects in different places. Because of all these steps, you can insert different steps in different places if you want to. You get the same signal molecule giving you two totally different effects in two totally different places. Okay, you don't have to memorize all of these. I'm not going to have you memorize these for a test. Now that being said, if there's an open no quiz, I might ask you something about that. But yeah. Anyways, okay, so. And just a few pKa substrates and talking about specific enzymes here. So we got muscle glycogen synthase, phosphorylase kinase A, protein phosphatase 1, pyruvate kinase, liver tyrosine hydroxylase. You don't need to know what all these are. You don't need to write them down. The point here is just that these are, there's a lot of different things that pKa can either activate or deactivate. This has a lot of effects. And which ones this is exactly working on is going to depend which ones are present in cells already, ready to go, ready to be phosphorylated. If some of these are missing, they don't get turned on because, hey, they're not there in the first place. So we have, again, because of this variety of things that you can get phosphorylated and turned on and turned off, you can get very different responses depending on which cells. And this is just a few. This is not even close to the complete list of pKa substrates. Are there any questions so far on the adenyl cyclase cyclic AMP pathway? Okay, so calcium channels and calcium. This is the second of those pathways that we were talking about. And so this involves a molecule called calmodulin. Calmodulin just stands for calcium modulated protein, as in it changes its function based on the presence or absence of calcium. And this binds binds for calcium ions. And once it does that, it becomes active. It can talk. I swear I can talk. I can say words with my mouth. Please. This is a relatively small protein. It's only about 148 residues long, so this, in the grand scheme of proteins, is not that big. Just to put in context, it's smaller than I am, which means it's very small. That was. It's all for okay. Context is everything. So here, this is a an example. This is kind of just a figure of calmodulin. You have kind of two different ends to it. You have these black balls are calcium. So you have two on one end, two that bind to the other end. Once it binds those those four calciums, it becomes active and it goes up and does this thing. So let's take a look at exactly how this works. Oh, I'm going to post up all of these diagrams. I meant to do that over the weekend, but then got kind of swamped, but I will get these up. The intention is today. But you will have access to these on D2L. Okay, so this starts out really pretty the same. You have a ligand that binds to a G-coupled protein receptor, or G-protein coupled receptor, which is bound to a G-protein, goes through the same thing here, where the G-protein, the alpha subunit breaks off as we replace GTP or GDP with GTP, and that goes off, and that's going to interact now with, instead of adenylyl cyclase, the difference here is we're interacting with a calcium channel. And that calcium channel is going to open and allow calcium to enter the cell. Once calcium enters the cell, now, I guess I should back up. This can actually be in several different places depending on how it's working. Sometimes this is actually in the cell surface and this calcium is coming from extracellular sources. Sometimes this is actually a channel on a organelle called the sarcoplasmic reticulum, depending on what cell type we're in, and we have a reservoir of calcium inside the sarcoplasmic reticulum that gets released into the cell as well. So it depends on what type of cell you are in, where this goes, what this does. Okay, but the calcium then gets into the cell. It's going to bind to calmodulin. Calmodulin can do a couple different things, which actually is kind of false advertising a little bit, because almost everything that we are talking about so far does a couple different things, and I've only been telling you one of them. But in this case, I'm going to tell you about two of the different things that does. Kind of the main line of things here is that can go on to activate protein kinase B or C, depending again on what cells that we're in, and then does pretty much the same thing that protein kinase A does. It phosphorylates an enzyme, which then either turns on or turns off that, and either starts turning substrate into product or stops that. Another thing that can happen here is that calmodulin can go and activate phosphodiesterase, which will then go break down cyclic AMP. So actually, this pathway can inhibit other pathways that are requiring cyclic AMP to work. Cyclic AMP is also smaller than I am. Are there any questions on this so far? Yeah.
Say the alpha subunit turn on the calcium channel and the
Adenylyl cyclase?
Yeah.
Yes. In some cells, it can do both things. So,
it turns off the cam and
it can. Yes. Again, well, okay. So, and this is a long story, but a lot of things are kind of sloppy in biology, and you get things like one thing you have genes that like get turned on. You make a bunch of transcripts just to break all that transcript down again. Like some of the most highly expressed genes in your body are just like the mRNA is just broken right down again. Like biology does all this weird stuff where things they get the job done. It's not always in the most efficient way, and as long as the inefficiency isn't so great to cause the like a selective, like an adaptive fitness penalty, then cool, everything's good. Just recycling stuff, like we don't worry about that, you know. Don't worry about the man behind the curtain that's just doing all kind of crazy stuff back there. We're gonna turn on this and turn this off. That being said, that's not always the case. Sometimes it won't turn on both of those. It depends on what cell, but there are some cells that yes, they will turn on both the dental cyclists and over calcium. Well, then finds calmodulin. But sometimes actually, to make this even more complicated, every time it opens a calcium channel, there's other pathways that this calcium can go to than actually finding calmodulin. So that's not always the case too. So each step, there's actually several different things that can end up happening. And I'm trying to give you a couple of them. I'm showing you a couple different steps here. Over the course of this, I'm giving you a couple different things that alpha subunit can do. Talk about a couple things that beta subunit can do, but this becomes wildly complicated and wildly fast. But yeah. Good question, though. Yeah.
So then, this calmodulin, is its activity dependent on the concentration of the calcium in the cell, or could there be more or less calmodulin because it's coming from someplace else, it's being generated from someplace else?
So yes to both of them. Yes, it is dependent on the amount, the concentration of calcium in there. Because in the end, I mean, everything is concentration dependent anytime you have an interaction between two molecules. It depends on what's the likelihood of those two like colliding and binding, right? So that is dependent. However, that being said, yes, you can also have varying concentrations of calmodulin as well that either have more or less activity happen as well. Yeah. So yes, both can happen. Any other questions? Okay, I'm going to leave that one actually there for a moment and not talk a lot more about that. But we're going to go on and talk about the next one, which is phospholipase C, the IP3 and DAG pathway. And this is just to give you, so I'm just giving you a handful of different pathways that come up. There's actually many. This is a few of the bears. Whoa, where are you going? Go back there. This is just a few that have been well studied for the G protein coupled receptor system, but there's a lot more. So, in this, we have phospholipase C. Phospholipase 3 breaks down phosphatidylinositol 4,5-bisphosphate, or just PIP3, which is a lot quicker to say. Now, it breaks down PIP3 into inositol 1,4,5-triphosphate, and we also get a 1,2-diacylglycerol out of that, or DAG. So IP3 and DAG. So let me ask you, what do you think this is? Can anyone give me a basic idea of what phosphatidylinositol 4,5-bisphosphate is? What's that? You said that? You said? Did you say it, Joe? Corbin? What did you say? Someone guess a phospholipid. It is! Ooh, it's a phospholipid. Okay, so phosphatidyl. Okay, what do you think the head group is? Inositol. Good job. Okay, it's inositol. And what do you think there's anything special bound to this inositol? Phosphate. How many of them? Two. Okay, cool. So actually, if you look at it, how many total phosphates are bound to it? 3, because you have to have one binding it back to the glycerol, right? Okay. Hey, good stuff. The names make sense sometimes, even if they're long. They kind of tell you some things. Okay, so when we chop that off, when we chop off the inositol 4,5-bisphosphate along with this, I don't know if the tittle is phosphate or not. But anyways, the phosphate plus that, then you get inositol. You get the inositol, and then a bunch of phosphates attached to it, 3 of them. And then you get the diacylglycerol. Diacylglycerol is just glycerol with 2 fatty acids that have been attached to it. So let's take a look at what this looks like. Once again, here's our diacylglycerol. This is the tail portion of this. On this glycerol, we have the phosphate attached. Here's the inositol with the 2 other phosphates attached. All phospholipase C does is goes snip right there, and then we get our diacylglycerol, so our two fatty acid chains here with the glycerol, and then we have our inositol triphosphate. So we have our inositol with 1, 2, 3 phosphates. So that's fun. We all enjoy doing that. Let's talk about how this works now. So again, we're going to start off very, very differently than we did before with a ligand binding to a G protein coupled receptor that's attached to a G protein. Okay, exactly the same as we started out before. And then we're going to replace the GDP with the GTP, and the alpha subunit is going to bind. I know, suspense. It is like you never know what's going to happen next. And this is going to bind to phospholipase C. Phospholipase C then attaches to our so sometimes it's called PIP2 and sometimes it's called PIP3. So if there's a PIP and it has a number after it, here we're calling it PIP2. Use that. We're going to bind that and then cut that off, and then our inositol triphosphate is going to, or IP3 is going to come loose and go do its own thing, and then our diacylglycerol is then going to go off and do its own thing as well. We have two different effects of this. So one of those effects is the IP3 here. Talk about the IP3 can come down here and open up a calcium channel in an endoplasmic reticulum. That's similar to what we were talking about before. And then once this calcium gets out, it can phosphor or activate a protein kinase C. And that protein kinase C is going to be free floating in the cytosol. And it can do something. Alternatively, the diacylglycerol up here, it stays in the membrane. So this phosphoinositol bisphosphate is a membrane phospholipid. Is going to that diacylglycerol is going to stay in the membrane. Is going to go and activate a protein kinase C, which can also go and do various things. Any questions with that, Howie? Yeah.
It's like kind of the same thing.
It's kind of the same thing.
Is the one that's in the membrane can leave the membrane?
No, this one stays in the
So
membrane.
the stuff comes in.
Yes.
Okay, cool.
And it's generally going to have slightly, this is not going to have the same effects as this one actually. So when I talk about protein kinase A, protein kinase B, protein kinase C, that's actually a whole class of kinases, not like one specific kinase that's going to do one specific thing. So they have slightly different effects. Any other questions?
Yeah.
Yes.
Are the two paths, like the DAG path and the PIP3 path, are they related or not, or?
They will tend to have related in that you end up eliciting a coordinated response. They're going to be part of the same coordinated response in the cell. Especially since you're going to get like an equal molar amount of both of these created. And so you're going to get approximately the same amount of this PKC and this PKC activated. Does that make sense?
Yeah.
Yeah.
For studying purposes, do you want us to like know the entire pathway for the test, like for this one and other ones, like other pathways like the calcium?
Yes, you should know the entire pathway. Any other questions? Okay, so let's talk about some ignored parts of this system so far. The beta, gamma, and subunit. It's not just hanging out there not doing anything. It also has effects. We're not going to talk about them by and large because I'm only trying to track down a few good examples of pathways in this system. I could sit here and teach an entire class on G protein second messenger systems because there are tons of them. But the beta gamma unit has effects as well. So that can also activate and inhibit intracellular effectors as well. One common thing that they do is they can activate potassium channels. They can also inhibit calcium channels. Now these are common things. These are not the only things that they do. They can also have some effects on adenylyl cyclase as well. They can also have some effects on phospholipase C. So the beta gamma subunits are not just sitting there, wishing that they got involved more. They do things. But this is just another way of kind of illustrating that at each point along this path, it's kind of like a very branching network of things that's happening, not just necessarily a linear chain where this follows this and only leads to this and only leads to this. Look at your skateboard over the AC. What's up with that? It's freezing. It's so wonderful. It's so hot in here. Okay. Okay, let's talk about a couple more. So we've talked about the G protein second messenger systems. That's not the only way in which we can get things happening. We also have a thing called protein receptor kinases. And these are cool. I like them. We're going to talk more about these a little bit in the future, but for now, these are involved in lots of different things, including things such as cell growth, development, differentiation. These are generally acting through changes in gene regulation. So these tend to have some sort of effect on the inside of the cell that's going to work its way into the nucleus and change the expression of some sort of gene, which will then have its effect that way. So there's actually a lot of them. There's more than 50 million humans. Actually, I think this number has gone up quite a bit in the last few years. And they tend to, what they do is they do something kind of interesting. They will phosphorylate tyrosine residues on themselves. So they phosphorylate, they're an enzyme that phosphorylates itself. And they tend to phosphorylate tyrosines, which makes that kind of encapsulates the name right there. They are a receptor that will phosphorylate tyrosines on themselves. And then what happens is that certain other proteins with a specific name called the SH2 domain can then dock with these phosphorylated tyrosine residues. And those will become either active or inactive. So those get turned on and turned off. One example of this that is fairly well known because of its impacts on human health is the insulin receptors. Okay, so this is how this tends to work. We have a receptor tyrosine kinase here. It binds to its ligand. When it binds to a ligand, it will then bind to another receptor tyrosine kinase that's also bound to its ligand. They will bind together and cause a dimer. And then these N-terminal chains will then phosphorylate each other. They will, so for instance, there's one on the left, will then phosphorylate the one on the right and vice versa. They phosphorylate a string of tyrosines on the intracellular side. Now in the case of the insulin, now from here, I'm going to talk specifically about how the insulin receptor works. So once this happens, you have a protein with an SH2 domain that can bind to it. In this case, it's IRS1, which stands for insulin receptor substrate 1. And the IRS1, in turn, is going to bind to and activate phosphoinositol 3 kinase, which then converts PIP2 that we've been talking about. Okay, so I screwed up earlier in the lecture. PIP2 to PIP3. Going to convert phosphoinositol bisphosphate to phosphoinositol triphosphate. And phosphoinositol triphosphate is then going to bind to phosphokinase B, or protein kinase B. I keep calling it phosphokinase. Protein kinase B. In turn, protein kinase B is going to phosphorylate glycogen synthase kinase 3. Glycogen synthase kinase, then, once it's phosphorylated, it stops acting by phosphorylating glycogen synthase, which normally turns off glycogen synthase. And so this will start working again. Normally it doesn't work. So the end result here is if you get a ligand binding here, glycogen synthase is turned on. Because through this, we are inhibiting the thing that turns it off. Does that confuse you yet? Another thing that also happens is that the insulin receptor substrate 1 will also activate a GLUT4 channel.
So what's happening GSK3 with the red
This?
cross out there?
So normally this inhibits this. Yeah. By being phosphorylated, it's inactivating glycogen synthase kinase 3. So it can no longer deactivate glycogen synthase. Does that make sense?
Yeah.
Yeah. You're turning something off to keep it from turning off something else. Yes?
So this, the insulin receptor is an example.
Yes.
This entire process is something to know.
Yeah, you should know this example.
Okay.
Yeah. It's like if you see someone going to turn off a light and you're scared of the dark, so you tackle them. And by doing that, you keep the light on. You didn't actually turn on the light yourself, but you can basically say you turned on the light yourself. I will demonstrate if someone tries to turn off the light. Anyways, so here's another example in which you can have in a cascade, you can have two different effects that end up doing two different things. But they're both coordinated. So from the IRS1, we get this pathway which then turns on glycogen synthase, which turns glucose into glycogen. And that's happy. We're storing energy. We're taking basic energy molecules and storing them away. We also get a process by which we open up glucose channels that allow glucose to come into the cell so that we have a substrate for glycogen synthase to make glycogen. And because this is diffusion that's happening here, you need to have a low substrate inside. So actually, by this converting the glucose into glycogen, that means our glucose levels actually in the cell stay low, which means that this glucose continues to pour in. We don't build up. We keep a concentration gradient there. So this is two different ways in which we can have two different effects through one signal cascade that have a coordinated response some situation. Are there any other questions? We're going to talk about one more real fast. The receptor serine threonine kinases, which are kind of similar. These are receptors, and instead of phosphorylating tyrosine, they're going to phosphorylate serines and threonines. And you have two different kinds of receptors. You have type 1 receptors and type 2 receptors. And then when the ligand binds, the type 1 and the type 2 come together. So it's similar to the tyrosine receptor kinases, in which we have multiple ones that are going to bind a ligand coming together. In this case, it's slightly different because the type 1 and the type 2, they bind it together, one single ligand together, but they come together. And then the type 2 receptor phosphorylates the type 1 receptor. Okay, so we have type 2 receptors, which for whatever reason T's and Y's like to sit on top of each other now, and the type 1 receptor, and together they bind some ligand. This is going to be, for instance, TGF-beta, and they bind it together, and they come together as they bind, in which case the type 2 will phosphorylate the type 1, in which case they bind a substrate. In this case, something called sera, which then binds our SMAD, which then is phosphorylated. These actually have kind of cool names that I would talk to you about a little bit. Because MAD is Mothers Against the Decacephalon, I think. Anyways, it's people naming these originally had some fun with naming them. We're just going to talk about it. Our SMAD, once it gets phosphorylated, then leaves and binds to SMAD4, which then SMAD4 goes into the nucleus and begins affecting transcription. Are there any questions on that? Yes?
Does it always form like the tetramer where it's like there's orbital and stuff in it? There's you got the two type 1s that are next to each other, and there's type 2s that are on the exterior or something.
Ask your question one more time, sorry.
So, like if the RTKs, if it's just two subunits coming together, it seems like it's 4 subunits.
Yeah, so in some cases, I think it's normally 4, is you have 2 type 2s and 2 type 1s.
Would both the type 1s and the outside, both sides get a phosphate then added?
Yes, they both get phosphorylated. Any other questions? Okay, thank you very much. I will talk to you on Wednesday. Yes, Nate.
Monday is fine.
Yeah, it's fine. No worries. We've got changing your major. Probably department chair, David. So what's your major right now? Biochem. And you want to teach bio?
Yes, I do. I haven't graduated to this lesson anymore.
Okay.
So, yeah.
Okay. So I would say the person that we best talk to is David Lindsay. Yeah.
Alright, so I'll reach out.
Okay. Cool. And let me know if that doesn't go anywhere. I will see what I can do.
I know I ask him a lot of questions. So.
Well, good.